Hey guys, so today I'll be discussing neural networks for bioinformaticians, and this will just be going through the basics, so a workflow with a specific example in mind. So what we're going to do is we're going to step through the process of performing dimensionality reduction and classification on a data set. So we're going to go through the choice of data set, so you understand the context, cleaning the data, filtering the data, selecting the features or pieces of your data set that you want to understand, normalization, and finally we get to the step where we can actually use a neural net to perform dimensionality reduction. We'll compare this to a common linear method, principal component analysis, PCA, and then we'll also show the same example. However, instead of the goal being to reduce the dimensionality in order to understand our data set, we'll be using a classification-based algorithm. So we'll be using, again, neural networks is one example, and another popular method called support vector machines. So the data set that we'll be looking at is clinical and exposure information for patients from the Cancer Genome Atlas, which is a really large resource and consortia of patient samples. And we've decided to stick to clinical and exposure information today because we have yet to go into biological data types such as gene expression and DNA methylation. And it kind of gives an intuitive example of what we're looking at. So as I mentioned, the goal is to use clinical features to identify different patient cancer types. So what I mean by this is, for example, say we have some clinical information about a patient, such as their age, demographic, and some exposure information such as their smoking status or drinking status, and other things like that. We can then ask, okay, knowing only these small bits of information, are we able to predict what cancer type they have, in the case of the classification example? And in the other example, are we able to understand these features better by performing dimensionality reduction? So as you can see here, I've just plotted some of the demographics, and we can see that it's not a balanced data set at all in terms of race. And there's also an imbalance when it comes to gender, however, it's marginal comparatively. And we can also see that there's an imbalance in regards to our goal, which is identifying the tissue origin of the cancer. So we'll have to look into each of these. So for data processing, which is the first part to understanding or to be able to perform any sort of analyses, we want to look at each column in the data set. So first we need to check, does it contain strings? Is it numeric? Are we missing data? So when we look at each column, we can then look at the possible features that we can then use for our later algorithms. So we could take demographic information, as I mentioned before, or exposure. One thing with data processing, in particular with the biological sciences, is that the data may not be what you expect. So in computer vision, where you have an image, it's quite easy to understand what you're looking at. Similarly, if you have, features from, for example, like whether someone likes a video or not, that's kind of a basic example that you get in other machine learning applications. However, with biological data, there's a lot of nuances with how different labs do things, so it's always good to check your assumptions. For example, if we take this data set, we can see that different people have inputted different names for tumor stages, so we need to combine them. For example, stage 1 or stage 4 here is just a 4 rather than actually having the stage prefixing it, so we need to merge those things together. Similarly, when we plot years smoked, we notice that things don't look right. So we can see if we print that out using our array. Also, I provide all the code to reproduce all the figures here, so you can follow along as well in the provided notebook. If we plot, just print out the values, we can see that there's string values and there's missing data, and they're not in the numeric format, which is what we expect for something like years smoking. So this is when your expectation doesn't meet the realities. That's why it's always important to check. So an example of how we process this is we can convert our years smoked by first replacing our string value, which was an odd character they decided to input, with NANDs, and then converting our feature, so pack years smoked, to a numeric data type. The next thing we need to be careful of, and we've already witnessed this once, is what do we do with missing data? So this is going to have a large impact on downstream analyses. So, for example, you could get missing values because of technical error in an experiment. You could get missing values because it doesn't actually exist. So it should be a 0. You could get missing values from all sorts of different causes, such as in the case of this clinical information, possibly the person who was taking the samples or talking to the patients didn't ask that information. So there's all sorts of reasons why you are missing data, and it's really important to understand the effect of imputing or filling in these values. So do we need to remove these patients? Should we fill in it with zeros? Should we use the mean value? Should we use a sample from the distribution about the mean? Or should we impute it using a much more complex algorithm? And to be honest, these questions are really important to think about before proceeding with your analyses, because, and also it depends on the problem that you're facing. So every data set will be unique. So unfortunately, you can't automate a lot of these procedures. The next thing that we need to investigate when we're looking at a data set is choosing the classes that we're going to actually look at. So as I mentioned, the goal of this video and the notebook that you've got was just an arbitrary goal to identify the tissue of origin for a cancer patient using just their clinical information and exposure information. So as I mentioned in the first slide, we have an imbalance in our tissue or organ of origin. We can see that, also there's some duplications as well. So tail of pancreas, NOS, body of pancreas. And so for my simple method, or my simple example, what I decided was I'm going to simplify it to the major tissue and pool endometrium or corpus uteri together, purely because my hypothesis was that mainly that would be dominated by females, and if I'm using clinical information, perhaps that would be something that I can extract. Well, we can see that it's still slightly imbalanced, so it seems to be dominated by lung cancer data set, but in the end, I decided to go with 3 classes for this example, mainly because I thought it would be simpler, and also because I thought that based on our features, for example, BMI, smoking status, etc., it might be easy to separate between these classes. The next part of, setting up a computational experiment for this sort of thing is choosing the features that you want to look at. So oftentimes, especially in the biological sciences, we're faced with way too many features in our data set that we want to understand. For example, I might be looking at a gene expression array or bulk or single cell experiment, and there's 20,000 genes that I want to understand. Most methods can't handle that, and most of the time that's too many features. So how do we choose the features that we want to look at? We can do this manually, which is what I'm going to show, or you can use methods to reduce or compress your features, which we'll actually look at in dimensionality reduction. One way of doing it manually is producing a pair plot, so essentially looking at the correlation and plotting the distributions of each of your variables, and then choosing which ones essentially look like they separate the classes out better in terms of class experiments. So basically looking at this, I thought that BMI seemed to provide a fair bit of information, smoke seemed to provide information, and so did age. So I chose these 3 features as the features to proceed with for this experiment. The question is, what do we expect the relationship to be between these data? It's always good to have a hypothesis. For example, if you have genes that you know about, you should develop a hypothesis about them. In the case of smoking and age, I predict that, the older someone is, the more likely they are to have years smoked. And weight, perhaps, the older you are, the higher your BMI, but I don't have much prior knowledge about that. And the next question is, will these have sufficient information to predict the cancer tissue type? So the next step is actually investigating the features that you've looked at a bit manually. So, for example, since we only have 3, we can just plot them together. It looks like they kind of overlap, but also there might be some information in there. And so another one of the reasons why I didn't stick with a traditional machine learning dataset such as iris dataset is because in biological sciences, when you're getting patient information, we never have these nice class separations, or if you do, you're very lucky. And another thing to note is, for example, pack years smoked. I imputed the data using zeros, and that's why we see this real not nice looking line down the bottom. It's where the patients were missing data, and I've made the assumption that that was because they didn't smoke. However, as I mentioned, it could just be because someone didn't sample from there. So these are important assumptions to state in a report or in any sort of thing. The next thing that we do, so the next step in our analysis of the data, or our data processing step, is to finally look at the correlation between our features computationally. So plotting them is one thing, but actually checking the correlation and checking these make sense. And this allows us to develop a hypothesis about what information is within the data set. And also, as a sanity check to make sure that things make sense. For example, BMI and weight are highly correlated. That makes complete sense. Age doesn't actually seem to be correlated too much to smoking, slightly, but there doesn't seem to be too much correlation between our features. So the first thing that we're going to do in terms of actually diving into it is to try and reduce the dimensions of our data set. So the goal is, can we reduce the dimensions of our data set to understand the relationship between our tissue types and these features a bit better? So just in terms of getting this into more, the mindset, so the number of features you have is the number of dimensions. I've kept it simple by just choosing 3. However, you could have potentially thousands of features. And so obviously that is far too difficult to plot visually. And while you reduce some information by plotting things naturally or reducing the dimensions, it also does add value, particularly in a system which you don't completely understand. So dimensionality reduction, how can we actually do this? How can we transform something from 3D to 2D? The most basic way we can reduce one dimension, but then we obviously lose lots of information. We could do a linear transformation, but how do we quantify this? And we could do nonlinear transformations as well. So the first thing to think about is, how do we quantify that a transformation is good? And we'll go through 2 ways of doing that, with our PCA and then our neural networks. So I just show you some example of linear transformations that are manually computed, and you can easily see which ones make more sense. However, these are not optimized with any heuristic. They're just, simple hypotheses about how our features can work together, and that's also a valid way of performing dimensionality reduction. So one of the most common ways that most of you probably would have heard about is principal component analysis. So what you're doing is you're changing the dimensions, so you're transforming your data to capture the components of most variation. So you actually have complete, if you have all your principal components, they fully explain all the variation in your data set. However, usually we'll cut it down to just the first 2 for visualization, or the first, n for however much variation you want to capture in your data set. So as you can see on the left, PC1 is the component of most variation, so you've got the biggest spread of the data along there, and PC2 is the next one. So it's pretty easy to visualize this, and then your data basically gets rotated to go along the axes of that dimension. However, when we do this to our data set of age, years smoked, and BMI, it doesn't look too informative. And so I guess the thing to think about is, why didn't our PCA just work? And part of that is because they capture the information in the input data, which means that if you don't normalize your input data properly, or you've got, outliers and things like that, that's going to be captured in the principal component analysis. A good way of, sanity checking whether your method or whether any method that you're using has worked is to, for example, in the case of dimensionality reduction, plot your input features or some known information onto the PCA. So here we can see that not much of the information in BMI has been captured. However, it looks as though our principal components are smoke, corresponds to our principal component 0, and our principal component 1 corresponds to age. So our question is this the right way to perform dimensionality reduction for our particular data set? Can we improve this? So one way we can improve this is by normalizing our data prior to transforming it. So there's a number of different normalizations you could do. So a common one in the biological data sciences is, for example, log2 transformation if you have count data. You could standardize your data, which means you transform your data to be 0 mean and unit variance, and you can scale your data. And scaling your data is really important for certain machine learning algorithms because of the way that the loss functions work, and we'll discuss that a bit more later. So log2 normalizing, so this is our input. We can see the x-axis goes from 0 to 100, and when we log2 normalize that, it goes from 0 to 7. And keep in mind that, you can't take a log2 transform or logarithm of 0, it's undetermined, so you'll get an error, so we usually add a pseudo count of 1 to all our values. You could also perform scaling, so you just take the minimum from the value and then you essentially divide it by the range of the data, and that transforms your exact same distribution, just between 0 and 1 instead. You could standardize your data, as I mentioned, and that's centering your data around 0 and then having unit variance. However, if you see what it's done to the smoking data, it hasn't actually made it that nice because we have such a heavily skewed tail of most of our patients falling into the what was 0 category or missing category. And lastly, you can combine these. So for example, I could do log2 normalization followed by standardization, followed by scaling our data between 0 and 1. So I'll just show you exactly what the effects of each of these are if you perform them prior to performing principal component analysis. So if we standardize our data before performing PCA, what we've done, rather than having BMI not be captured by our principal components, now we find that BMI is essentially our PC2. And our age has become our PC1, and our years smoked has become less dominant in our principal components. If we instead scaled our data between 0 and 1, we get back to the one we had before, which makes complete sense because it's following the same distribution. And lucky last, if we log transform our data followed by standardizing it before we perform principal component analysis, then what we've got is what you see here, which seems to be quite a nice capturing of each of the pieces of information that we're provided with or that we've input into our principal component analysis. So we've got BMI captured, we've got years smoked, and also our age. We can also compare this to the 3D plot of our BMI. Now we've got our normalized years smoked and our age on the z-axis. And we can see that each of them kind of corresponds to what we see in the PCA. So it looks as though it's captured a lot of the information. So one of the things to always check with your principal component analysis or dimensionality reduction is kind of like what features do they correlate with when you have a small set of features at least, and how do they correlate to things that perhaps were left out of the data set. So what do they capture other information that perhaps we didn't know about? So we can see that PC0 heavily anti-correlates with age and smoking and kind of positively correlates with BMI, and PC1 correlates strongly with smoking and BMI. And as you can see, by nature of principal component analysis, your principal components are orthogonal, which means they have 0 correlation. So now we'll change tact and we're going to do the same dimensionality reduction. However, we're going to use a neural network. So a neural network essentially has an input followed by a series or a single matrix, essentially multiplication, so some weights, which is tied to some biases and a nonlinear activation function or a linear one, so some sort of function. And then same thing, weights, biases, activation function on the output. And each of the parameters in the model, which are your weights and your biases, are learned. We'll go through that more in the next lecture, but that's kind of the overview of how it works. If you're performing dimensionality reduction, like I am in this very simple case, I'm going to go from 3 features down to 2 features, back to 3. So keep in mind with this sort of dimensionality reduction, just like with PCA, you aren't actually providing the model with the labels of the data, so I'm not telling it which of these inputs corresponds to which of our tissues. So if we do that architecture of a neural network, we train it and we use a linear activation function going to our encoding node. what we get actually in our autoencoder is very similar to our principal component analysis. And that's unsurprising because the, way to capture most variation in the dataset linearly is already almost fully explained by the components of most variation. This is also a good way to sanity check that your model is actually learning correctly. Like if you can't essentially perform PCA, if you've written a neural net yourself, you're probably not doing things right if you're using a linear activation function. So for another example, just to give you an idea of how these different activation functions work, if we used a sigmoid, which squashes our data between 0 and 1, this is what we get. So same information is captured, however, how our essentially plot looks is very different. And we can also see how our autoencoder features correlate with our principal components. If we use a rectified linear unit, which is another common activation function, which essentially squashes your data at 0 and then is linear, we can see again the effects of that. Capturing the same information, however, a different reconstruction. What we can see as well in the autoencoder and principal component correlation is actually that autoencoder component 1 correlates very strongly with PC0, and autoencoder component 0 anti-correlates strongly with PC1. So you are capturing the same information or similar information. So what is the limitations of neural networks? So they're limited by the number of features that you have compared with the number of data that you have to train. So we've got a very contrived, simple example with only 3 features. And how does this relate to PCA? So PCA is not going to be biased by how many features you have in your dataset. It's always going to find the components of most variation, whereas the neural network might find something completely arbitrary and might be noise. So it's also more susceptible, I guess, if you don't have enough data to like outliers and noise. But I guess it really depends on the system that you're trying to learn from. So if we move to a more complicated example, we can actually capture non-linearities as well. So in the previous one, we were just really showing an example of, one layer. So this is my dodgy drawing. I didn't draw in all the lines, but essentially rather than going from 3 to 2 to 3, what we've done, we've actually expanded our dimensionality out to 6 to 8, then back down to 2 to 8, and then to an output. And now we've got these nonlinear functions between each of these weights and the subsequent layers. When we do this, we see that our autoencoder actually, the output looks very much like it's not a linear transformation. Each of the previous ones looked quite linear, and that makes sense because we only had 2 layers. So we can see a bit more class separation now, but it's quite an interesting shape. And we again see, using ReLU activation and more nodes, that the features seem to carry similar information to in our original PC, but just a funky shape. So again, limitations of neural networks. Now that we've actually got many layers and nonlinearities, our data no longer conforms to a particular distribution or shape in the latent space. There are lots of different ways that we can actually train our model to follow certain distributions, but we won't go into this now. Okay, cool. So moving on to classification. So what we want to do is use the same features that we use for dimensionality reduction to predict the cancer tissue of origin. So we'll go back to keep in mind that we have an imbalanced data set. And the next thing we'll look at is actually, before we dive into doing this, do our features contain information that is able to separate? So the simplest way that we can validate that hypothesis is by performing a simple t-test and testing whether our features of interest, so our BMI, age, and years smoked, actually differ significantly between the 2 data sets, or 3 data sets, I should say. And we see that BMI does seem to be significant. Age contains relatively little information comparatively, and years smoked seems to contain somewhat information about the differences between the groups, which gives us kind of confidence that perhaps if we are learning from this, we are learning something important about the relationships between these data types and the tissues of origin of the particular cancer types. So the first method I'll discuss is a support vector machine. So just like PCA was performing a linear classification, not classification, a linear dimensionality reduction, support vector machines are pretty cool because they perform linear classification, however, in a higher dimensionality space than the original data. So what they do, rather than PCA, which uses a linear transformation to reduce the number of features, essentially, or combine them, the support vector machines transform your data through a kernel, so this function can be a linear function, it could be a nonlinear function, to a higher dimension such that the classes are able to be separated linearly. So as you can see from this example, we've used, I think, a Gaussian kernel on here, and we've then been able to separate between the red and the green classes by performing a plane as the decision surface. If we look at how well this separates our data set, it looks like it's very good at classifying lungs. So this is a confusion matrix, so there's many ways that you can quantify how well your machine learning algorithm has classified your data. And I personally like confusion matrices for this because you can see your true label and your predicted label. So my true label, for example, was endometrium, and I predicted it correctly in this one. My true label was endometrium, but I predicted it to be kidney and lung. So what we can see is when my true label was kidney, I actually got I was less successful at predicting the correct label than I was at predicting that it was part of the lung data set. So it's not necessarily good, but lung was quite good at predicting it, and perhaps that's because it's a dominant feature. Well, there's the most cases in our data set. So let's see how well this compares to our neural network. So I use the same architecture as I did in the previous example. So again, a very simple architecture using our 3 feature input down to 2 dimensions. For a neural network that's classifying, you don't need to do this. It could be a larger set of dimensions if you want to capture more information. And then it goes to an output where I'm essentially aiming to see, okay, is this kidney, lung, endometrium, or corpus luteoli? So I've used encodings to classify each class. I've one-hot encoded my data, which means that I have 3 outputs, one corresponding to kidney, where I'll be getting a 1 if it's kidney, or 0, so I shift my data through a sigmoid. Similarly for lung and endometrium. So the internal nodes, just to keep a thought out here, while I've got the same internal nodes and I can look at them just the same as I can look at them in the autoencoder example, they're not being optimized to reconstruct the input. That means if I actually plot them, they don't look particularly interesting or capturing lots of variation in the data set or anything like that. Because what this architecture is optimizing for, so the loss function for this, is using binary cross entropy. So I'm aiming to maximize the number of these ones on the end that I get correct. So if I train my model on that data set, what I get is 65% accuracy for a 3-class problem, which is not bad, slightly better than the support vector machine, given random would give us approximately 33%. And the question then becomes, is accuracy the best metric? And again, how does the balance of our classes affect our accuracy? So we see this time that, at least in our neural network example, and it'll change each time because they're they're non-deterministic, we see that, we get the most classes in each one correct. So I guess that's kind of good. So the question then becomes, how did this all work? So that we'll go into this a bit more in the next lecture, and we'll actually do a walkthrough of neural network and PCA and dive into what we're actually doing. So I'll see you all then.